being placed. So when I don't see it, what's happening is it's being focused right there on the optic disc. And then it is not being created.
Dude. Bam.
Isn't that weird?
That is so
Disappears right there.
I know.
I see it the whole way.
Do not drop anything.
That is.
So if you were to look into somebody's
No, no,
eye
no, no. It's
with
in between.
a tool called an ophthalmoscope,
I know.
this would be where the optic disc is. Remember I said the retina also has blood vessels?
Perfect
So
focus.
this is where the optic disc is, and there's blood vessels that actually come into the eye with the optic nerve. So if you look back at this diagram, you'll see it fits right here. So we have the choroid layer that has blood vessels, but the retina also has blood vessels. And so when you look at this image, those are the blood vessels coming in with the optic nerve. And then just medial to that, so the optic disc is always medial to the fovea. Notice how I drew it here going off to one side, so the optic nerve is going to be closer to the nose. Because we don't want to have our direct line of focus with the optic disc. You'd have this blind spot in the middle everywhere you look. Anyone doing a case study on macular degeneration? There's one person in the morning class. With macular degeneration, it refers to this area right around the optic disc. It's called the macula lutea. Macula means spot, and lutea means yellow. So there's this yellow region right around the fovea centralis. And in macular degeneration, it's a condition that increases frequency with age. It can be wet or dry, depending. It's a problem with leaky blood vessels. And it causes loss of vision, guess where? In the fovea centralis. So it's the most frustrating kind of blindness that I can think of, in the sense that you start to lose the ability to focus right in the center of your vision. You can see the periphery fine, but guess what? It's out of focus because the image is not being created on the periphery. So you see something that's out of focus, you move your eyeball to look at it, and then it becomes even more blurry, or even absent. It can be completely absent because of macular degeneration. So wherever you look, there's a blurry or a blank spot there, because those, right, the fovea centralis is right in the center of what we see. All right, so now let's look at the effect of the lens, the pathway of the light, and then how we focus that pathway. So before we do so, we need to identify some of the spaces in the eye. I'm going to leave this diagram up, but erase what I've drawn in the middle of the eye. So if you want a copy of that, go ahead and take a picture. And so as I identify the spaces, rather than draw this all over again, you can refer back to the copy of this image that I drew on Tuesday. Okay, so if you have that in your lecture notes, you can use that, but it's also in the diagram in your lecture notes.
Oh my God. Next week we'll have
Tell a lot about a person by their cell phone cover.
Okay, I'm basic black OtterBox.
What does it mean if we don't have a cover then?
You like living on the edge. All right, so we have two cavities that we look at with the eyeball. From the lens to the cornea is identified as the anterior cavity. And from the lens to the back of the eyeball is identified as the posterior cavity. Now we divide the anterior cavity into two chambers. So between the iris and the cornea is the anterior chamber. And between the lens and the iris is the posterior chamber. and we don't subdivide the posterior cavity. Remember the name of these structures here? The ciliary processes, the muscle is going to be in here, okay? So the ciliary processes function is to secrete the aqueous humor. And aqueous humor is what we find in the anterior cavity. It's made by the ciliary processes. So it fills up the posterior chamber, flows through the pupil. That's just the donut hole in the iris. Flows through the pupil and it's continuously made, just like CSF. So we have to reabsorb it back to the vascular system, and that occurs by leaving the anterior chamber through these vascular, kind of a combination of vascular structures and sinuses. And these are known as the scleral venous sinuses. Their older name, when they were named after an individual, was Canal of Schlemm. And it sounds too much like phlegm and gross mucousy stuff. So those are found in this point where the cornea joins the sclera, which is the limbus. So that region is known as the limbus. I can get it right here. So this is the limbus, and there's the scleral venous sinuses. Now, as I indicated, this is constantly being produced, constantly being reabsorbed. So if there's a problem in its reabsorption, it increases the pressure within the eye, and that can push back on the retina and result in damage to the nerve cells, known as glaucoma. Okay. Glaucoma is not curable, but it can be treated. My dad puts eye drops in his eyes morning and night. His dad, my grandfather, went blind with glaucoma. And so my dad's 83 now, and he wears glasses, but he's almost always had to wear glasses for nearsightedness, and he doesn't have the nerve damage from the glaucoma. So they're not exactly sure what causes it. Yes?
Oh, I'm sorry, finish what you were saying.
No, I was going to say, we don't know what causes it, but there are, so that it can't be cured yet, but there are medications that can help to increase the passageway of the fluid across the spaces. So these are almost the equivalent of the arachnoid villi, as far as the movement of the aqueous humor out. Yes?
In a diabetic-induced blindness, what exactly happened?
All right, so diabetes, the high levels of glucose, affects the surface of red blood cells, causes them to become sticky, and that affects their transport through the blood vessel walls, can cause them to clog or constrict. And so the blood vessels that you see on this image here, supplying the retina as well as in the choroid layer, become leaky, can constrict. There can be separation between the retina and the pigmented layer. And so throughout the body, our morning group did a case study on diabetic neuropathy, and you have some questions on your take-home on that. And the blood vessels, the capillaries become leaky, the smaller blood vessels constrict, and basically the organs or tissues lose a good blood supply. And so in the eye, the neurons there can die. You can get blindness from those dying neurons or from the retina detachment. With the autonomic neuropathy, neurons that are carrying autonomic, sympathetic and parasympathetic cannot function, so you lose autonomic function. Neuropathy is a very common, it's not the only reason my mom has diabetes type 2. And it started off with neuropathy, and that's about all she has so far, is they either have tingling or loss of sensations in the distal extremities. So they will often damage their feet, burns or stubbing their toe. Derek, one of the guys that did the case study on Tuesday, said he had a patient at the hospital where he works at that almost lost his foot as a result of going barefoot on the beach and not realizing that he had cut his foot. And then the second side of that problem is when you do get damage, you don't have a good vascular supply to repair the tissue. So you don't get the nutrients and the oxygen that you would need to repair it. So with diabetes, it's a loss of good blood supply to the nerves. And therefore blindness can occur. All right, so let's look at the structures that light is going to pass through. This is just an image showing the passageway of the aqueous humor as it moves through the posterior chamber and anterior chamber. So now let's look at the structures that light passes through. And light is going to, let's see, may I erase these here? Okay. So the first structure that light passes through is the cornea. And then it's going to enter the anterior chamber and pass through the aqueous humor. From the anterior chamber, and then the pupil, and then the posterior chamber. And then it's going to pass through the lens. And then it's going to enter the posterior cavity, and this is a jelly, very much like the consistency of grape jelly. You'll see it tomorrow. This is known as the vitreous humor, or you may also see vitreous body. And so each of these structures has a density that's different than the other, and a density that's different than air. So when light waves pass through structures of different density, they are bent. So this next image shows what we see if we look at a spoon in a glass of water. So here the light waves coming off of this are passing through the air. Here they're passing through the glass. And here they're passing through the water and the glass. So you can see it looks like it's broken as it's bent from different degrees. And this is why we talked about using a drop of oil if we were using the oil immersion lens, because the oil has the same bending degree and capability as the glass does. They're very similar. So it's easier to keep the light waves parallel and in focus. So all of these structures that I have lines beside, the cornea, the aqueous humor, the aqueous have a C. Seems like it should. I don't know. This doesn't look right. The aqueous humor, the lens, and the vitreous humor are all going to bend light to some degree. Only one of them can vary the degree to which it bends the light. And that would be the lens. So you guys remember the term we use for that?
Refraction.
So refraction is bending. So all of these are going to be able to do refraction. Okay, all of them are going to be able to do refraction, but only the lens is capable of accommodation. Meaning it can bend the light a little bit or it can bend the light a lot. And that was the role of the ocular motor nerve and the ciliary muscle, changing the amount that the lens is bent. Okay. So now I would like to erase the entire board. Anybody need pictures? You guys have that? screen. And let me make this image a little bit larger. When we create an image on the eye, it's kind of like the microscope. It's upside down and reverse left to right. Okay, so that's why what you're seeing here. When we look at something that's close up, this is what we talked about with the oculomotor nerve. So here the arrow is being focused on the retina, and ignore the fact that it's on the optic disc here because it's actually on the other side of it. So the arrow is now upside down. Okay? Object that is farther away does not need to be, the light waves do not need to be bent as much to place the image on the retina. Okay. As we move closer, therefore then, as we move an object closer, the lens needs to round up because it needs to bend that light wave to a greater degree. And that's why when someone has, they get older and they have to wear reading glasses, it's because their lens has lost its elasticity and is not capable of rounding up. It's flat and it gets a little bit rounder, but not real round. So you can see out here, but not up here. All right? You're the one with the birthday, my dear. Not that the rest of us don't get them either, but all right. So, which of these would we want to use, a convex lens or a concave lens if we want to bend the light waves towards each other? A convex. Okay? So what do we mean by convex? If you have difficulty remembering this, so our light wave is going in this direction.
An arch versus a dip.
That would be convex. Okay? Whereas a lens, you walk into a cave, is going to be shaped like that. All right? So this is a convex lens. It will take the light and converge the light waves, bring them together. Whereas a concave lens will take the light and diverge, spread the light waves apart. Okay? So now when we look at this, as we talk about nearsightedness and farsightedness, keep this in mind. Do we need to spread the light waves apart? If we spread the light waves apart, is it going to increase the focal distance or decrease the focal distance? It's going to increase it, okay? Because they have to come together and we've made them farther apart, so it's going to take longer distance for them to come together. If we bring them together first, then it's going to take a shorter distance as we bend them even more. All right. So I'm going to erase this and put up a typical eyeball and retina. So here's what happens normally, okay? So normal vision. We have our lens. We have the retina. We're going to start out without any change necessary. We have an image over here that we're bringing our light from. And the light waves come in and are bent so that they come to a focus. Let me do this right place here. So they come to a focal point on the retina. Okay, that gives us a focused image. Right. Now, if I bring an object closer, it's going to be bigger. It's not going to be bigger, but it's going to appear bigger. It's going to take up more of my visual field than it would if it was 100 feet away. So if we bring that object closer, let's say this is the object here. We bring that object closer, it's going to take up more of my visual field. Does that make sense? So my hand out here is going to appear bigger when it's up here. It's going to be this size versus my visual field versus out here. So we're going to have to bend the light wave more to actually get them to focus on that retina. If my image is farther away, okay, they're already, by the time it gets to my lens, they're already close together. It won't have to bend it quite as much. Does that make sense? All right. So now let's look at what happens to the lens for each of these conditions. I'm going to describe what has to happen. Now I use a different color. So normal vision without bending it very much. Okay? So this is normal. What do I have to do to the shape of my eye, my lens, if I need to increase the bending? Make it rounder. So if I want to increase the bending, a greater degree of convergence needs to occur, all right, then I make it bigger, and I end up with the image still on my retina. I mean, I make the lens rounder. So I have a greater degree of bending as it comes through. So what I've done, just using one, is I've taken this and then bent it to bring the lens inward, to bring the light inward. Okay? Now, because I'm very far away, and I'll redraw this one down here, if I'm very far away, I don't need to bend it as much. So my lens is going to be flatter. This is what my own eyeball does. My lens is flatter, and I don't need to bend the light as much to get it into focus. All right? So for close-up viewing, the lens becomes more round. For distance viewing, this is with a healthy eye, the lens becomes less round or more flat. Now, I don't know how many of you, getting farther and farther, have used a regular, okay, even regular is the wrong word, an SLR camera where it actually has a lens that you adjust, all right, rather than a cell phone camera. So when you adjust you're looking through a camera with a lens attached to the front of it, and you turn it, you're moving it closer or farther away from the film. That's the equivalent of rounding up or flattening the lens as you change the focal distance. Okay? Now let's say that we have a problem with our lens, or we have a problem with our eyeball. Okay? So let's say that, may I raise the board? So someone needs corrective lenses. If they are nearsighted, what happens, here's their lens, here's their retina, thank you. That's with the eyeball, that was the wrong word. So typically when they are nearsighted, when they look at things up close, everything's fine. Okay? When they look at things far away, they're out of focus. What happens is either the eyeball is too long, or the lens is can't flatten enough, and essentially the image comes into focus in front of the retina. Of course, the light waves don't stop there, they keep on going. So by the time they hit the retina, the image is out of focus. Okay? So you can think of nearsightedness as the image is created too close to the lens, or you can see things close up. All right, but you can't see things far away. So what would we need to do to correct for this? Do we need to bring the light waves closer together, or do we need to spread them apart?
Rather apart.
We need to spread them apart. So we use concave or convex? Concave. All right. So we put a concave lens in here ahead of the normal lens. What we end up doing is light comes in, is spread out before it hits your lens. So it's diverged before it hits your lens, and then your lens, you bend it, and it would come into focus on the retina. Okay? So the corrective lens is spreading apart the light waves before they get to your lens, because your lens isn't capable of keeping them far apart, or your eyeball is too far away for that to work. Your eyeball is too long. So that would be nearsightedness. So this would be myopic. So the image is created at a focal distance in front of the retina, and it's corrected with concave lenses. Myopic. Nearsightedness. Yes.
Slightly off topic, but in color blindness, where does that occur? Does that occur in the brain? Or
That occurs,
on
no,
the
it occurs
eyes?
with the cones, and we're going to talk about that after break. It has to do with, we have genes for 3 different color cones. There's actually multiple genes for greens. And so those are carried on the X chromosome. And so predominantly color blindness is seen in men. And it's usually for the green or the red that there's a problem.
Stoplight colors.
Yes. Okay, so hyperopic, all right, is farsightedness. So if that's nearsightedness, can I change this now to farsighted? Can everybody see what I have up there for farsighted, for nearsighted? Okay. So now let's look at farsighted. So people who are farsighted, my kids were early on, so like when they started school, a couple of them had to wear glasses for farsightedness. I'm nearsighted, but as people tend to get old, as people get older, the lens becomes less capable of rounding up, so they cannot do close-up vision, and so they're considered to be farsighted. They may not have normal vision far away, but neither do they have normal vision close up. So for a while in my 40s, my vision actually got better because I was becoming more farsighted and I was already nearsighted. So it started becoming a little bit better, and I then finally had to start. I have a farsighted lens in this eye and a nearsighted lens in this eye. Now my husband does that too, but there's such a difference between his nearsighted and farsighted vision that if he were to wear a farsighted contact that would allow him to read small print, it would be such a big difference between the correctedness in both eyes that he would notice it just looking around. So he still has to wear reading glasses if he wants to read really fine print. All right, so let me go through this and then. So farsighted means you cannot see things close up because the lens cannot round up enough. That's what happens with age, or the eyeball may be too short. So what happens with normal vision, without correction, the light waves cannot be bent enough, and so if we extrapolate, the vision focal distance would be beyond the retina. So again, by the time the light waves reach the retina, they have not yet come together in a focal point. So in this case, the corrective lenses need to bend the light waves more. So you can see here, without correction, if we extrapolate the focal point, it would be beyond the retina. And so we use convex lenses. To bend the light waves a bit before they get to your lens, because your lens can't bend them enough. Okay. So now we would bend these even more.
Bless you.
And now your lens, with that correction, is capable of focusing them on the retina. Okay? So I have, like I said, I have far-sighted contacts in this eye. I can read, I can even read smaller than that, but I can read the tiny, tiny print on a medicine bottle. I'm looking for something here. So this is easy for me to read. Conformed to ASTM, non-toxic, etc. Is there a tiny print on that one?
Yep.
Okay. So distributed by North American Coffee Partnership, purchased, please recycle for best taste, drink, whatever. So I can read that tiny print down there. Now if I close this eye, I can't even make out a letter. Okay? So I was doing all that reading with my left eye. Now we have these vision tests that you can use today or tomorrow. You're supposed to read them from 20 feet. Each of these blocks is about a foot long. So if I close my right eye, I'm reading with my close-up vision, and I told you I have to close, I can't blink it shut. So I can read E and H and N, and then D maybe, F and N, and after that, if I'm guessing, it looks like maybe an F or a T, a Z, and who knows what the last one is on the fourth line. Okay? So now if I close my right eye and use my distance vision, the bottom line looks like an N, an F, maybe an X, T, Z, F, and H. All right, so that was a very, very bottom line. So those are what you're seeing here. So this bottom, the top line, the best one I could read here was this one, because this one I don't, maybe this one a little bit. 20 over 100. All right, my vision is the top number. So it means 20 over 100 means that when I stand at 20 feet, I can read what somebody with normal vision could read if they were standing at 100 feet. So 5 times that distance, someone with normal vision would be able to read the H and the N. Okay? So my number is the top number. The bottom number here says 20 over 15. So again, my vision is the top number. I was standing at 20 feet. But at 20 feet, I could see with my correction contacts what somebody with normal vision would have to walk up to 15 feet to see. So when the bottom number is smaller than the top number, you have better than normal vision. When the bottom number is bigger than the top number, then your vision is getting poorer. Okay?
I can't see anything. Go ahead.
So this is just another slide that's illustrating emmetropic means normal vision over here. Myopic is nearsighted and hyperopic is farsighted. And okay, let's take a break. When we come back, we'll do physiology. We'll be back to action potentials and the pathway. It'll take about 30-35 minutes or so, and then we'll be done with that. Go back. Is that one? Okay. Yeah.
Are we gonna have an intermission before the lab?
Yeah.
Like how long? 10?
15 minutes.
Sweet.
So come back in maybe 10 minutes because we didn't go that long this time. We'll have a little bit longer time for lab.